Welcome to The Explainer. Today, we're diving into the microscopic world to uncover the computational magic behind one of the greatest puzzles in modern biology. I'm talking about predicting protein structures. We're going to look at exactly how scientists use mind-blowing computing power and strict structural rules to literally see the unseen.
So let's start right at the fundamental challenge here. How do we actually know a protein's 3D shape? At their core, proteins just start out as a simple, one-dimensional sequence, just a string of letters. But in biology, shape is everything. That 1D sequence has to fold itself into a highly complex, highly functional three-dimensional shape just to do its job. Figuring out exactly how it gets there? Well, that's the ultimate puzzle. And this beautifully illustrates our end goal. Take a look at this intricate, squiggly, folded 3D ribbon. It might look like a tangled mess, but actually, every single twist and turn is precisely dictated by the laws of physics and chemistry. Translating that flat 1D sequence into this magnificent 3D structure is a massive, giant puzzle. And today, we're going to solve it together using 3 different computational strategies.
Here's our roadmap for this explainer. We're climbing a computational difficulty ladder today. We'll start with level 1, homology modeling, move up to level 2, fold recognition, and finally tackle level 3, ab initio methods.
All right, let's kick things off with level 1, homology modeling or copying from a known template. Think of this first method as having a massive advantage because, well, you basically get to use a cheat sheet. You're using a previously known protein that's really similar to your unknown sequence. But there's a catch. There is a strict threshold to use that cheat sheet. If you look at the curve here showing sequence identity versus alignment length, there's a very clear divide. To confidently say our sequence is going to fold like the known template, we need a sequence similarity of at least 25%. If we fall below that curve into the gray area, those alignments carry practically zero information about the 3D structure. So, if you don't have that 25% match, this tool just won't work. But, assuming we do have that 25% match, here's how this homology approach actually works, broken down into 6 crucial steps. First, we find a set of related sequences. Second, and honestly this is the key right here, we align our target sequence to those known templates. Third, we construct the 3D core backbone. The vital thing to remember is that the strongly conserved regions give us the conserved structure. Where the structure diverges, well, we have to use predictions to fill in the gaps. Fourth, we construct the flexible loops. Fifth, we model the location of the side chains using what's called a rotamer library. And finally, step 6, we optimize and verify the model to improve its likelihood. Now, hold on, we can't just trust that step 6 blindly. These models aren't automatically correct just because a computer spat them out. No way. They must strictly obey the real laws of physics and geometry. We have to make sure the carbon backbone conformations are allowed according to a Ramachandran map. We need legal bond lengths and angles. Peptide bonds absolutely have to be planar. Hydrophobic, meaning water repelling. Residues need to be safely tucked away in the right environments. And crucially, there can be literally 0 physically impossible holes inside the 3D structure. Basically, the whole shebang just has to make physical sense. Moving right along to level 2, fold recognition, also known as threading the unknown. So what happens when we don't have that 25% match and our sequence has no obvious twins in the database? Well, we rely on a backup plan. Let's compare these 2 techniques side by side so you can really see the trade-off. Homology modeling gives us incredibly high accuracy, but it's rigidly restricted by that 25% similarity rule. Fold recognition, on the other hand, casts a way wider net. It can be applied in so many more cases, but it sacrifices some precision, making it generally a bit less accurate. Why is that? Because instead of having a clear template to copy, fold recognition relies heavily on the mathematical accuracy of a scoring function to basically guess the best fit. Here's how that threading process works. Our sequence of amino acids has all sorts of different chemical properties, right? Some are hydrogen donors, some are acceptors, some are hydrophobic. You get the idea. We literally thread this input sequence onto a massive library of known shapes, or folds. Then we calculate a score for the compatibility between our sequence's chemical properties and the physical reality of that structure. We just keep testing different known folds over and over until we get a hit. We're looking for a structurally sound arrangement that gives us a good score.
And that brings us to level 3, ab initio methods, the ultimate boss battle.
Guys, this is truly the ultimate boss battle of computational biology.
Imagine a scenario where we have 0 templates, no known folds that fit, absolutely nothing to go on except the raw, flat sequence of amino acids. Ab initio is a Latin phrase meaning from the beginning, and that is exactly what we're doing here. We are building a protein structure entirely from scratch. Now, this method relies on 2 massive assumptions. Assumption 1, all the information about the structure of a protein is contained entirely within its sequence of amino acids. And assumption 2, the structure that the protein naturally folds into is the one with the lowest free energy. It inherently wants to find its most relaxed, physically stable state. But to actually pull this off, scientists need an incredibly complex toolkit. First up, we need a robust scoring function, a potential, to evaluate the energy of our models. Second, we need a massive, and I mean massive, search strategy. We have to mathematically model real physical forces like van der Waals forces and Coulomb interactions. And because the number of possible shapes is practically infinite, we use probabilistic search algorithms, things like simulated annealing, to generate and test vast ensembles of candidate structures, all to find the one with that perfect, lowest free energy.
A really prime example of this toolkit in action is the famous Rosetta algorithm. And let me tell you, Rosetta doesn't mess around. It uses a fragment library derived from known structures and applies that simulated annealing process we just talked about. To get a reliable prediction, Rosetta will generate roughly 1,200 candidate structures. And get this, for each of those structures, it crunches through 100,000 independent cycles of optimization. 100,000! It is an astronomical amount of computational heavy lifting, all designed to zero in on the perfect fold. So just looking back at the whole journey we've taken today, we start with a simple, flat string of letters on the left. That's our raw amino acid sequence. And by scaling this computational ladder, whether we're copying a template in homology modeling, threading the sequence in fold recognition, or just battling it out from scratch with ab initio algorithms, we end up with a beautifully complex, highly functional 3D structure on the right. To wrap things up, I want to leave you with a thought about where this is all heading. We just saw how computationally intensive predicting a single protein from scratch can be. But as our computing power grows exponentially and these algorithms get even smarter, will we soon be able to predict any protein in mere seconds? Just imagine a world where understanding a new disease's protein structure happens instantly, potentially unlocking life-saving cures overnight. It's an absolutely thrilling frontier in computational biology, and I hope today's explainer gave you a strong foundation to understand it. Thank you so much for focusing in and learning with me today. Keep questioning and keep exploring.